So now we're going to look at the cell. So the cell is considered as a basic unit, the basic functional unit of all living organisms. So the cell is capable of independent functions. So the cell can perform all these functions on its own, and that's why it's considered as an independent functional unit. Even though cells are dependent on one another, a cell is a full package, meaning the cell can work on its own. So we are saying the human body develops from a single cell known as a zygote. But we know that zygote is a product of two cells, that is a sperm cell and a female egg cell known as a oocyte or ovum. Then from there, those cells begin to multiply in number until an embryo is formed, until the fetus is formed, until a full human being is formed. After birth, this human being continues to grow and develop. So basically, there are two types of cells. In biology, we have prokaryotic cells as well as eukaryotic cells. So I'll not focus on that. I'm sure by now you know the difference between the two. Let's look at the components of the eukaryotic cells. These are cells in which we find human or animal cells. So our cells belong to eukaryotic cells. So this discussion is about the components and the functions of eukaryotic cells, to be specific, human cell. So the cell is composed of two parts. Mainly, we can divide the cell into the nucleus, into the cell membrane, and the cytoplasm. Now we know that inside the cytoplasm, we find various organelles. So the cytoplasm can be divided into two parts: the cellular organelles and the cytosol, which is a fluid in which these organelles are bathing. So this fluid is jelly-like in texture. It's not like water. It is more of a jelly-like substance. So basically, this is a diagram showing us the cell. So we have the cell membrane. Then inside here, we have cytoplasm. Then we see the bluish part here, that is a cytosol. Then we have organelles of various shapes and different colors here with different functions. So we start with the outer part of the cell, which is a cell membrane, also known as a plasma membrane. Okay, so the cell membrane forms a barrier between, or a boundary between, the internal environment and the external environment of the cell. So it divides these two environments, an internal environment and external environment. Apart from that, the cell membrane creates a semi-permeable barrier, meaning it allows certain molecules of a particular size and charge to enter the cell. At the same time, inhibits certain molecules of particular size and charge to enter the cell. So there's that semi-permeability. So let's look at the components of the cell membrane without wasting time. The cell membrane is composed of the following: phospholipids. So these phospholipids create a double layer. So there's this outer layer and there's that inner layer. Then we have proteins, labeled blue here. Then we have carbohydrates or polysaccharides, which are sugars. Here they are shown to be green. So we have these green molecules attached to protein or the phospholipids. Then we have cholesterol. Here it is shown to be yellowish. So we have cholesterol there. So to be specific, there are about 4 basic molecules found within the cell membrane. That is number 1, phospholipids, number 2, cholesterol, number 3, proteins, number 4, polysaccharides. Now, let's look at the structure of the phospholipids. So we'll start with the phospholipids because of how they influence the cell membrane. They give us a double layer structure. So this is a structure of a phospholipid. So phospholipid has a head and a tail region. Now in the tail region, we have two fatty acid molecules. So this is a head region made up of a nitrogenous compound, a phosphate bridge, and a glycerol. Glycerol is more of a sugar. So this is a glycerol, that's a phosphate bridge. Then up there we have a nitrogenous compound. So the head is said to be polar. So the head is polar because of nitrogen and phosphate there. So we have a positive and a negative charge, whereas the tails is non-polar. Even though there are two molecules of fatty acid, the tail is non-polar because these two molecules are not charged. A fat is neither positive nor negative, and these two are fat in nature, and that's why it is a non-polar region. The tail is non-polar. Apart from that, if you observe, you see that one of the tails is more straight, like this one. The other one is bent or kinked. So the one that is kinked or bent is made up of unsaturated fatty acids, whereas the one that is straight is made up of saturated fatty acids. So we see that saturated fats, these are fats when at room temperature, they are in an oily state or liquid state. For example, cooking oil, when it is left in the house, you see that it is in liquid form. So it is made up of unsaturated fat. It is made up of saturated fat, sorry. Then unsaturated fats at room temperature, they form solids, like for example, wax. Okay, so any fat that is left at room temperature, you find it that it's hardened or condensed, then it is made up of unsaturated fat. So that's as a basic application. So here, head, they are polar because of the negative and positive charge created by the phosphate and the nitrogen found in the head, whereas the tail is non-polar. So this is a single molecule of a phospholipid. Now, on the cell membrane, we have plenty of these. And we see that the heads and the tails face in different directions on the cell membrane. So this is the head, these are the tails. Then here we have the heads and the tails. So since the tails are made up of fatty molecules, whereas the heads are made up of nitrogen and phosphate, you find that the heads, apart from being polar, the heads are said to be hydrophilic. They interact with the water molecules that is found here in the internal environment. At the same time, the heads that are in the outer layer here, they will interact with water molecules on the external environment of the cell. Since the tails are made up of fatty substances, they shun away from water molecules, and therefore they are said to be hydrophobic. So the tails will meet in the middle of the lipid bilayer. This is one layer, that is another layer. Then in the middle here, that's where the tails are found because of the nature of their chemical properties. The heads interact with water, hence hydrophilic. Tails shun away from water molecules, hence hydrophobic, and they will be found in the middle of the lipid bilayer. So already this creates a semi-permeable barrier. There's this barrier here created by the tails of the outer layer. There's also another layer of semi-permeability created by the tails of the inner layer. So water molecules that are inside here will not cross these tails. They can interact with the heads, but the moment they reach the tails, the water molecules are repelled back inside the cell. Same with water molecules found on the outside of the cell membrane. They can interact with the heads, but the moment they reach the tails, the water molecules are repelled back to the external environment of the cell. And that way, the fluid inside the cell and the fluid outside the cell is maintained at a constant concentration because of this semi-permeability created by the cell membrane. Okay, so that is one part of the cell membrane, phospholipids. Here it's just written to be lipid, but actually they are phospholipids because of the phosphate group that we saw to be found in the head region. Now let's examine another part of the cell membrane known as cholesterol. So cholesterol is found closer to the tails of the phospholipid molecules. So cholesterol, it acts more like a seal. So it fills up all the gaps that are left in between the phospholipid tails so that there is no possibility of water molecules to leak through any cracks left within the cell membrane. So cholesterol makes the cell membrane to be more flexible, more stable, and more semi-permeable. Prevents movement of substances across the cell membrane. So any small gaps that are left in between the tails, they are filled by cholesterol molecules. And we see that the cholesterol molecule is similar to a fat molecule. Okay, now a cholesterol has a head and a tail. Instead of having two tails like a phospholipid, it only has one tail. Now, we observe that the whole cholesterol molecule is non-polar. There is no part that is polar and part that is non-polar. The whole molecule is non-polar, meaning it is neutral. It has no positive or negative charge. Now, if you observe the tail, you see that the tail of the cholesterol molecule is bent or kinked, similar to what we saw in one of the tails of the phospholipid molecule. So even the tail of a cholesterol molecule has unsaturated fat. Okay, so this is the structure of cholesterol. Then we look at the protein molecule. So if we observe the protein molecule, we see that there are so many different types of proteins found on the cell membrane. So here they are labeled in blue. So I want you to observe this one and this other protein molecule. So on the cell membrane, we find about 3 types of protein molecules. Number one is that we have an integral protein. This is a protein found within the cell membrane. It is found within the 2 layers of the cell membrane. Then we have transmembrane protein. So like this one and this one. These are molecules, these are proteins that are crossing both layers of the cell membrane, the inner layer and the outer layer. And that's why we call it transmembrane, because it crosses the membrane. Then we have peripheral proteins. Peripheral proteins are proteins that are only attached to one layer of the cell membrane. For example, this one is attached to the inner layer of the cell membrane. We can also have another peripheral protein attached only to the outer layer of the cell membrane, hence the name peripheral, because it is located on the peripheral region or far away regions. So basically, there are 3 types of proteins on the cell membrane. Integral, found within these 2 layers. Transmembrane, crossing the 2 layers. Then we have peripheral, attached only to one layer of the cell membrane. Now we look at the functions of proteins found on the cell membrane. If you observe this one and that one, you see that one protein has a pore, a gap in between. Then the other one is solid. So let's begin with the one with the gap. So the one with the gap is known as the channel protein. Channel protein. Now, channel proteins are used for transportation. Channel. So this channel will allow transportation of substances or fluid from the inside the cell to the outside and vice versa, from the outside to the inside of the cell. Now, channel proteins are of various types. So they are channel proteins that can only be accessed by charged molecules. So we call such as ion-gated channel proteins. Ion-gated. Ion is an electrolyte that is dissolved in water. So ions are either positively charged, we call them cations, or negatively charged, we call them anions. So for example, for this protein, if it's an ion-gated channel protein, for this channel to be opened, a substance that is trying to enter the cell should either be a negatively charged substance or a positively charged substance. That's when this molecule, this protein, will allow transportation of that molecule. If it has not charged, then this protein cannot be used for transportation of such kind of a substance. So we call such as ion-gated. So the gate, for it to open, the molecule has to be either negatively charged or positively charged. Then also have what we call ligand-gated channel proteins. Ligand-gated. Ligand means a certain substance, for example, a hormone. So those in physiology or anatomy, they are called ligands. So a ligand-gated channel protein will only allow transportation or movement of a substance across it if it is a hormone, if it is an enzyme, or a factor. There are certain substances that we call factors in the body. They are not hormones, and they are not enzymes, so they are known as factors. For example, growth factor. Okay. Now, a ligand-gated channel protein will only allow passage of a ligand here, a substance that is classified as a ligand, which is a hormone, an enzyme, or a factor. Okay. So those are the types of channel proteins that are found on the cell membrane. So a ligand-gated channel protein cannot transport an ion. An ion has to pass through an ion-gated channel proteins. Now, if you observe this one, it has no passage, but it is a solid protein. So we call this as a carrier protein. So a carrier protein, unlike a channel protein, a carrier protein has binding sites and usually has 4 binding sites. So 2 on the outside. So there will be 1 binding site there, another one here, that is 2. 2 on the inside, 1 here, another one there. So that is 2 inside. So total is 4. So a carrier protein will transport 4 molecules in total. 2 molecules will be transported from inside the cell going out. 2 molecules will be transported from outside the cell going in. Now, what happens is that when the binding sites are saturated, 2 are attached inside and 2 are attached outside there. What follows next is that this protein will change its shape, what is known as conformation change or morphological change if you want. Then the outer region will bend and move towards the inner region, whereas the inner region will move in the opposite direction from the inner of the cell to the outer of the cell. And that way, substances that were attached on the outer part of this protein will move inside the cell, and substances that were attached to the inner part of this protein will be transported to the outside of the cell. Now, for this change to occur, we need energy. So a carrier protein uses energy to undergo conformation change and transport its substances in and out of the cell. Unlike a channel protein will only allow transportation of 1 molecule at a time. So if 1 molecule is going out, we cannot have another molecule going in at the same time. It's not possible. They have to give chance to each other. But here, on the carrier channel, on the carrier protein, transportation is simultaneous. 2 molecules in, 2 molecules out. So that's a function of proteins found on the cell membrane. Mainly, they are used for transportation in such a manner as I have explained. Now we look at the last part of the cell membrane known as the polysaccharides. So here they're shown as green. So there are those polysaccharides attached to protein and polysaccharides attached to the phospholipids. Now polysaccharides are carbohydrates, like I said. So they don't exist on their own. They are not independent. They are always attached either to lipid or protein. And that's why they are given the following names. The polysaccharides attached to the lipids, they are known as glycolipid because they form now a compound molecule, a molecule composed of carbohydrate and a lipid, hence glycolipid. Polysaccharides attached to the protein, they are given the name as glycoprotein because they form one molecule. Now it is a carbohydrate and a protein, hence the name glycoprotein. Now we see that on the inner surface or the inner layer of the cell membrane, we do not have these polysaccharides. They are only found on the outer surface of the cell membrane. Now, since these polysaccharides project outwards, they face away from the cell, you find that they will form a layer which is known as a glycocalyx. So here we see some spikes, more like fuzzy area on the cell membrane. This is a cell membrane. That is another cell membrane there. Now, there's a layer of the cell membrane that is composed purely of carbohydrates. For example, here, this is a carbohydrate attached to a lipid. So this is a glycolipid. Now, if you draw a line from here and there, you see that this layer from there up to here, it is composed purely of carbohydrates. So this is what is known as a glycocalyx. Okay, so the glycocalyx is a layer of the cell membrane composed purely of carbohydrate or polysaccharides. And we see that the glycocalyx is important for various functions of the cell. So we see that any substance or cell or any particle that wants to interact with the cell membrane must first of all interact with the glycocalyx. And that's why now we're saying the glycocalyx is the first layer of the cell membrane through which various cells will interact with that particular cell. So among the functions of this glycocalyx is cell recognition. So glycocalyx or carbohydrates on the cell membrane act as the cell markers or cell surface markers. These are receptors. Apart from that, they help in cell attachment. So attachment of one cell to the next is facilitated by the same glycocalyx that we have mentioned. Apart from that, we have heard of people having various types of blood groups. No, I'm blood group A, I'm B, I'm AB, I'm O, and all that. What are we talking about? What we're talking about when discussing blood group is simply the type of sugar that we have on our red blood cells. So a red blood cell is like a cell, like any other cell of the body. It has a cell membrane as well, meaning it is going to have a phospholipid, it's going to have proteins, it's going to have cholesterol, it's also going to have polysaccharides. Now, the polysaccharides on the surface of the red blood cells are important because they enable us to identify different types of blood groups. So, if you are O, it means the type of sugar you have on your body is O, for lack of a better term. If you are B, it means the type of sugar you have on your body on the red blood cells is, on the surface of the blood cell is B. If you are blood group A, it means the type of sugar you have on the surface of the red blood cells is A, sorry. Now, what that means is if the red blood cells in your body have A as their sugar, it means they're going to produce antibodies against other types of blood groups. And that's why A and B blood types cannot mix. That's why you do blood grouping and cross-matching. Make sure that the type of sugar in the blood that you're about to give this patient, the type of sugar found on the surface of red blood cells in this blood that you're about to give the patient matches with the type of sugar on the surface of red blood cells that the patient has. And that way there will be no reaction. If you give two different types of blood groups, this sugar on the surface of these two different blood groups will cause a severe reaction. There will be rejection of these two blood types because each blood type is producing antibodies against another type of blood, and that way there will be a serious reaction that may result in death. So that's all about the cell membrane. In summary, it is composed of the following: phospholipids, which have a head and a tail. The heads are said to be polar, whereas the tails are said to be non-polar. On top of that, the heads are hydrophilic, the tails are hydrophobic. Now, such kind of a molecule is known as amphipathic, a molecule that is having both polar regions and non-polar regions. So a phospholipid is an amphipathic molecule. Apart from that, we have cholesterol. Cholesterol fills up the gap, like we can see here. It found in between the tails of the phospholipid molecules, and we said it makes the cell membrane to be more fluid, more flexible, and more resistant to movement of cells across the cell membrane. So it makes it more semi-permeable. Next is that we have proteins, which are not shown here. We have proteins here, and we mentioned the types of proteins found on the cell membrane and their functions. We have integral proteins found in between the two phospholipid layers. We have transmembrane proteins crossing both layers. Then we have peripheral proteins found attached only to one layer of the cell membrane. And finally, we have the glycocalyx, made up of polysaccharide chains that are attached either to the phospholipids, giving us glycolipids, or attached to protein, giving us glycoproteins. So, function of the cell membrane. Number one, it creates a barrier between the internal environment and the external environment of the cell, then controls movement of substances in and out of the cell. So that is known as selective permeability. Because of the presence of the glycocalyx, the cell membrane regulates cell-to-cell interactions and cell-to-microorganism interaction. Apart from that, the cell membrane provides identification of a cell because of the type of carbohydrates found on each and every cell in the human body. So that is known as a human antigen. These are carbohydrates found on the surface of our cells. Apart from that, cells are able to identify foreign antigens, foreign cells, and even cells that have been altered or undergone some mutation, or they've become cancerous because of the changes that occur on their cell membrane. The sugar that we talked about, the carbohydrate, will undergo some change, and that way the cell will identify to say, the body will identify which cell has undergone changes. So the next lecture will look at the internal aspect of the cell, which is divided into two parts, namely cellular organelles and cytosol.